So last article, we talked about abiotic and biotic factors. Abiotic meaning that there are factors in our ecosystem that are non-living, such as sunlight and water and soil. And then you have your biotic factors, which are living things, such as animals and plants. Today we're going to talk about what is population. So I'm going to start reading the article under what is population, and you can follow along. In everyday life, we often think about population as the number of people who live in a particular place. New York City has a population of 8.6 million people, or Monowi, Nebraska has a population of just 1. Just think. You could double the population of Monowi if you felt like moving there. In ecology, a population consists of all the organisms of a particular species living in a given area. For instance, we could say that a population of humans lives in New York City and that another population of humans lives in Gross. We can describe these populations by their size. What we often mean by population when we're talking about towns and cities, as well as their density, about how many people per unit area, and distribution, how clumped or spread out the people are. So if you think about a city like Chicago, the density is going to be more packed. It's going to be more dense because there's a lot more people living in Chicago in a smaller area. And if you think about your hometowns or Woodstock, the density, it's going to be less dense because we have less people living in a larger area. Next, we're going to talk about demography, describing populations and how they change. In many cases, ecologists aren't studying people in towns and cities. Instead, they're studying various kinds of plant, animal, fungal, and even bacterial populations. The statistical study of any population, human or otherwise, is known as demography. And remember, guys, that the prefix dem means people, just like in democracy. So you can remember that's the study of population, a human population. Why is demography important? Populations can change in their numbers and structure. For example, age and sex distribution for various reasons. These changes can affect how the population interacts with its physical environment and with other species. By tracking populations over time, ecologists can see how these populations have changed and may be able to predict how they're likely to change in the future. So, for example, everybody should have gotten a census card in the mail. The government is taking a census, which means they are taking all this information about the demography of the United States. How many people live there? How many females are living in the United States? How many males are living in the United States? What age are they? So they're able to use that information so they can plan and change the living spaces or maybe shopping around town so they can better accommodate the people's needs. By tracking populations over time, ecologists can see how these populations have changed and may be able to predict how they're likely to change in the future. Monitoring the size and structure of populations can also help ecologists manage populations. For example, by showing whether conservation efforts are helping an endangered species increase in numbers. In this article, we'll begin our journey through demographics by looking at the concepts of population, size, density, and distribution. We'll also explore some methods ecologists use to determine these values for populations in nature. Population size and density. To study the demographics of a population, we'll want to start off with a few baseline measures. One is simply the number of individuals in a population, or population size. Another is the population density, the number of individuals per area or volume of a habitat. Size and density are both important in describing the current status of the population and potentially for making predictions about how it can change in the future. Larger populations may be more stable than smaller populations because they're likely to have greater genetic variability and thus more potential to adapt to changes in the environment through natural selection. So if you had a large population of animals, let's say squirrels, we see a lot of squirrels around the area that we're in, they are going to be more successful at continuing their population and their genes. If you compare that to a super small population, let's say of endangered animals, those animals have a really hard time increasing the population of their specific species and having their genes passed down. A member of a low-density population where organisms are sparsely spread out might have more trouble finding a mate to reproduce with them than an individual in a high-density population. So measuring population size. To find the size of population, can't we just count all the organisms in it? Ideally, yes, but in many real-life cases, this isn't possible. For instance, would you want to try and count every single grass plant on your lawn? Or every salmon in, say, Lake Ontario, which is 393 cubic miles in volume? It's huge. Counting all the organisms in a population may be too expensive in terms of time and money, or it may simply not be possible. For these reasons, scientists often estimate a population size by taking one or more samples from the population and using these samples to make inferences or educated guesses about the population as a whole. A variety of methods can be used to sample populations to determine their size and density. Here we'll look at two of the most important, the quadrant and the mark recapture methods. The quadrant method. For immobile organisms such as plants, or for very small and slow-moving organisms, plots called quadrats may be used to determine population size and density. Each quadrat marks off an area of the same size, typically a square area within the habitat. A quadrant can be made by staking out an area with sticks and string or by using a wood, plastic, or metal square placed on the ground, as shown in the picture below. After setting up quadrats, researchers count the number of individuals within the boundaries of each. Multiple quadrat samples are performed throughout the habitat at several random locations, which ensures that the numbers recorded are representative for the overall habitat. In the end, the data can be used to estimate the population size and population density within the entire habitat. So using the given picture in your article, you can see it doesn't have to be a large plot of land that they're setting up the quadrant around, but it's just a small piece of land. And in that little square that they have there, they're counting all the specific organisms. And then they're gonna, around the community, they're gonna plant several more of those in random spots and do the same thing. So then they're able to get a kind of really good reading on how many species are in that habitat or that community. The mark recapture method. For organisms that move around, such as mammals, birds, or fish, a technique called the mark recapture method is often used to determine population size. This method involves capturing a sample of animals and marking them in some way. For instance, using tags, bands, paint, or other body markings as shown below. They do a lot of this with great white sharks. I think there's a website that you can go on and track all the great white sharks and kind of see where they're traveling to and where they breed. So that is really cool. So they take this approach with animals that are moving a lot. Then the marked animals are released back into the environment and allowed to mix with the rest of the population. Later, a new sample is collected. This new sample will include some individuals that are marked, recaptures, and some individuals that are unmarked. Using the ratio of the marked to the unmarked, scientists can estimate how many individuals are in the total population. Example, using the mark recapture method. Let's say we want to find the size of a deer population. Suppose that we capture 80 deer, tag them, and release them back into the forest. After some time has passed, allowing the marked deer to thoroughly mix with the rest of the population, we come back and capture another 100 deer. Out of these deer, we find that 20 are already marked. If 20 out of 100 deer are marked, this would suggest that marked deer, which we know are 80 in number, make up 20% of the population. Using this information, we can formulate the following relationship. So this approach isn't always perfect. Some animals from the first catch may learn to avoid capture in the second round, inflating population estimates. Alternatively, the same animals may preferentially be retrapped, especially if a food reward is offered, resulting in an underestimate of population size. Also, some species may be harmed by the marking technique, reducing their survival. The approach also assumes that animals don't die, get born, leave, or enter the population during the period of study. Alternative approaches to determine population size include electronic tracking of animals tagged with radio transmitters and use of data from commercial fishing and trapping operations. Species distribution. Often, in addition to knowing the number and the density, so how many in a certain area of individuals in an area, ecologists will also want to know their distribution. Species dispersion patterns or distribution patterns refer to how the individuals in a population are distributed in space at a given time. So you think about, are they all clumped together? Are they all spread out evenly? Are there pockets of different groups of these animals spread out evenly over a specific habitat? So those are all things to think about. The individual organism that makes up a population can be more or less equally spaced, dispersed randomly with no predictable pattern, or clustered in groups. These are known as uniform, random, and clumped dispersion patterns, respectively. So as you guys can see on your article, there is a visual of the three ways that animals are dispersed throughout a habitat. The first one is uniform dispersion, and you can see how those red dots are evenly distributed through the box. They're evenly spaced. The next one is random dispersion. You can see how those are kind of randomly placed throughout the box. Some of them are clumped together, or some of them are further away from one another. But you can see that there's kind of that random pattern there. And then the last one is clump dispersion. You can see in the box how those animals or species in that box are clustered together in their own little groups, and each group is pretty much away from one another. So the first one, uniform dispersion. In uniform dispersion, individuals of a population are spaced more or less evenly. One example of uniform dispersion comes from plants that secrete toxins to inhibit growth of nearby individuals, a phenomenon called allelopathy. We can also find uniform dispersion in animal species where individuals stake out and defend territories. Random dispersion. In random dispersion, individuals are distributed randomly without a predictable pattern. An example of random dispersion comes from dandelions and other plants that have wind-dispersed seeds. The seeds spread widely and sprout where they happen to fall, as long as the environment is favorable, has enough soil, water, nutrients, and light. And last one, clump dispersion. In a clump dispersion, individuals are clustered in groups. A clump dispersion may be seen in plants that drop their seeds straight to the ground, such as oak trees, or animals that live in groups of schools of fish or herds of elephants. Clump dispersions also happen in habitats that are patchy, with only some patches suitable to live in. As you can see from these examples, dispersion of individuals in a population provide more information about how they interact with each other and with their environment than a simple density measurement. So the summary. In ecology, a population consists of how many species are in a specific habitat. The statistical study of populations is called demography. Two important measures of populations are population size and population density. So, how many people are there or how many species are there? That's population size. And population density refers to the amount of species or the amount of people in a certain area. The number of individuals per unit or volume. Ecologists often estimate the size and the density of population using quadrats. Remember those things you can make with string and wood boards that are kind of randomly sampled throughout a habitat, or the mark and recapture method, which is used more widely with animals that are constantly on the move, such as fish, birds, and larger mammals. A population can also be described in terms of the distribution or dispersion of the individuals that make it up. Individuals may be distributed in a uniform, random, or clumped pattern. Uniform means that the population is evenly spaced. Random indicates random spacing, and clumped means the population is distributed in clusters.